In the field of conservation biology today, there has been one predominant mindset of how to conserve and protect species that are either on the decline or are potentially right at the cusp of becoming extinct. That mindset has been to target this species, let's say, for example, the American bison. It had been reduced through a number of different factors, but primarily due to commercial hunting for their hides and due to habitat destruction. Now what happened was, at the cusp of their extinction, we were able to protect them from any further hunting, create preserves, and breed them to the point where today, from only 500 individuals being reduced from over 60 million across all of North America, we were able to bring them up today to over 500,000. Now, today, it's a success. We were able to take a species that many thought would have gone extinct, and we were able to propagate them to the point where now they are a viable population which is self-sustaining. Their gene pool is sufficient enough where a disease would not affect all of them, and to the point where they would be at risk of becoming extinct. However, this is not the case for all animals. Take the Javan rhinoceros as an example. There are 70 individuals in a singular park on the island of Java in South Asia. Now, from that, you could theoretically breed back a population, get them to be self-sustaining, and potentially recolonize some of their range that had been lost. The problem with that being is their gene pool has been severely reduced. With only 70 individuals, if you were to restore them to even, let's say, half of their prehistoric range, their gene pool would be so small that a disease could rapidly affect them and wipe them back out to the point where they are today, or potentially even completely into extinction. Now, that you can see the effects in other species today where that's happening. Take the koalas in Australia, where not only are they at risk due to the fires happening currently today, but they also have a chlamydia that has been transmitted through many of them that they are not genetically prepared to face. The same thing happens with the marsupial predator, the Tasmanian devil. There are two strains of a transmittable cancer that have been spreading throughout the island. Now, they wiped out already 95% of their entire population. Now, if nothing is done, they may too go extinct. Now, you may be wondering, how can we stop this if simply breeding them and protecting them is not enough? Emerging in this field is the idea of artificial genetic diversity, and that is to basically take the eggs of devils and in a laboratory using genetic editing tools such as CRISPR-Cas9, edit their genomes either by a little bit or through any variable amount to the point where you can artificially recreate genetic diversity into their population. Now that would both help in bringing their numbers back up, but it would also help them to prepare against diseases in the future where they'd be less susceptible to it, as they'd have more of a genetic barrier to prevent it. However, this technology does not end there. Take their recently extinct larger cousin, the thylacine or Tasmanian tiger. This was also a predatory marsupial that lived in modern times, at least, in Tasmania. However, it at one point ranged all the way from New Guinea through the mainland to Tasmania, as mentioned. With this species, there is enough DNA in pickled specimens and frozen remains in laboratories all over the planet that they have been able to create genomes that are almost complete, very little breakage between them. Now, all it would take would be to get a fully functioning genome of the species, just one, edit it into a relative, let's say the numbat, which is its closest, to create a functioning genome of the species. Now, by doing so, again with the same technology, you can then edit that genome into a variety of them with a wide range of genetic diversity to not only taking it a step further from, let's say, the devils, and creating an entirely new genetically diverse species from one that is extinct, all from a singular genome. By using this technology more in this field, not only can we conserve populations with an abundance of genetic diversity that are on the decline, but we can also save those that have a very lack of it and prevent them from facing extinction as well.